Hello everyone, I would like to thank the organizers for inviting me to speak at the Virtual BTS Congress. My name is Delian and today I would like to show you how using probabilistic thinking and taking uncertainty into account can help us make the right decisions. And we need to make the right decisions in drug discovery because it costs billions to bring a new drug to market and it can take years. Now let's begin with an example of a very important decision. Suppose you're moving to a new house and you want to be close to a hospital because you suffer from a heart condition. How do you make a good decision as to which house to choose? House A is located in the town center and its average ambulance arrival time is shorter compared to House B located in the countryside. However, you want the ambulance to arrive before 10 minutes because research has shown that for your heart condition it's really important to be seen by a doctor or a paramedic within 10 minutes. Now, if we had information not only about the point estimate of average arrival time, but the arrival times of multiple instances, and we could build a whole distribution of arrival times, we would be making a better decision, a more informed decision. If we look at the graph now, House A, which has a shorter average arrival time, is located in the town center and often suffers from traffic. So ambulances don't make it within 10 minutes 30% of the time. House B, on the other hand, is located in the countryside, doesn't suffer from so many traffic issues, and ambulances don't make it in less than 10 minutes only 16% of the time. So if you wanted to buy a house where the ambulances will get to you within 10 minutes, you would be wise to choose House B over House A. There are two main sources of uncertainty, the data that we collect and the models that we fit to our data to try and understand the relationships that are happening and to predict the future as to what's going to happen in the clinic. For example, if we're running an IC50 assay in cells and we're trying to predict a clinical outcome such as drug-induced liver injury, our IC50s would be determined with a specific measurement error. Some of our validation compounds may be misclassified. If we're using a threshold to determine if a compound is active or inactive, for example, a compound that is 29.9% active will be labeled as inactive, and a compound just over the threshold, 30.01, will be labeled as active. That would introduce its own uncertainty and is usually discouraged. If our measurement we're trying to make is below the limit of detection, it may end up as a missing measurement. The model brings its own sources of uncertainty. What is the distribution that we're choosing to model the data? Is it counts and then we'd use the Poisson distribution? Should we be using the normal distribution? If there are outliers, maybe we should consider using the T distribution. If we know that the data is supposed to be strictly positive, we can use a version of the normal called truncated normal. When we have ordered categories, we can use ordered categorical. And the parameters that we have in the model, for example, the weights for the different measurements that we have chosen have their own uncertainty. In the drug discovery world, we want to make the right decisions. So incorporating all of those sources of uncertainty is really important. It's important because drug discovery is a long process. Drug discovery is an expensive process, and it can often feel as working at the edge of chaos. It feels this way because the space of drug-like molecules is absolutely vast. It's estimated to be 10 to the power of 60th. That's more than there are stars in the universe. So it can feel very overwhelming. It can make it slightly less overwhelming if we look at the largest chemical database, which only has about 110 million. And in the AstraZeneca collection of small molecules, we have around 2 million molecules. Now, from this vast chemical space, and all the molecules registered in PubChem, we have about 12,000 small molecules registered with some kind of human activity as drugs. So really, the properties that make a good drug are very rare in the chemical space. This brings me to one of the greatest challenges we have in early drug discovery, and that is of class imbalance. We are screening millions of compounds against a single target, but only a small fraction are active. And if we do a back of an envelope calculation, let's say we had about 1 million compounds, and we had an assay that is really specific, 99.9, and really sensitive, 99.9 sensitivity. But we're expecting out of the whole collection a very small percentage of compounds to be the real actives, let's say 1 in 1,000. After we've run our assay on the whole collection, we end up with about 2,000 hits. Of those, only about 50% would be true hits and 50% would be false positives. Due to the vast majority of inactive compounds, this really specific and really sensitive assay delivered us a hit list with 50% false positives. Now you'd say, well, that's easy. These random errors can be mitigated by just running a confirmation screen. And that's true. If you run a confirmation screen on those 2,000 compounds, only about 50% would confirm, which is about the average confirmation rate we expect from an early HTS screen. While these stochastic or random errors are easy to triage, there's a whole set of errors that are systematic and assay artifacts that are much more difficult to get rid of. These sources of interference can be compound specific. For example, multiple academic and industry groups have come up with these lists of pan-assay interfering compounds, frequent hitters or nuisance compounds that involve a large group of different players like redox cyclers, compounds which are contaminated with different metals and can interfere with assays, compounds which aggregate and can interfere with the measurements, also compounds that quench the fluorescence if you're measuring fluorescence endpoints, compounds that are fluorescent themselves or precipitate. If you're doing cell-based assays, these are compounds which have unspecific cytotoxicity, the genotoxic or the disrupt tubulin, and they often become frequent hitters in the different assays. And here I would really recommend the Darlin review that has recently come out in Cell Chemical Biology. It's a really excellent review that shows you what kind of strategies you could implement in your screens to triage those assay interfering nuisance compounds and to derive high quality hits. But the other very important source of systematic error is plate effects. For example, if you have a plate pattern, maybe the cells on the edge are less healthy than the cells in the middle of the plate. Or if you have a batch effect, maybe two batches have been seeded with a different cell density, and you can end up with assay results that are quite different. So you need to find ways to account for interference coming from compounds as well as interference coming from plate and batch effects. And so far I've given you mostly theoretical underpinnings of uncertainty and sources of errors, and with the next few slides I would like to illustrate it with a couple of examples from the drug-induced liver injury field and from trying to determine safety of compounds in our portfolio based on in vitro results. The first example I would like to share with you is a high content imaging assay we use in HepG2 cells designed to predict human drug-induced liver injury after 24-hour incubation with different drugs. So the assay is truly multiparametric. It measures multiple different phenotypes. For example, the cells are stained with HUX for the nuclei, TMRM for the mitochondria, TOTO3 for cell death, and with NBDP, a fluorescently labeled phospholipid analog, to check for phospholipidosis. Based on those stainings, we can determine 8 different phenotypes. The cells are either healthy, they're hypopolarized if they're still alive, but their mitochondria have been hypopolarized, hyperpolarized when the mitochondrial membrane intensity is really high. The cells could be dead, or there's this dead cytoplasmic phenotype where there are early signs of cell death. When they round up, we label them as rounded up apoptotic, and based on the green phospholipidosis staining and the spots, we label those compounds as having phospholipidosis or not. And what we measure in this assay is the first phenotype that departs from our untreated population. So we're looking at the most sensitive phenotype, and we're looking at the first move from the healthy state into something else. We have tested over 400 compounds with DILI annotation in this assay, and we have tried to summarize the results in this scatter plot. So each dot on the scatter plot is a compound. If the compound is liver toxic, we've labeled it red. If it's not, it's labeled with blue. And on the x-axis, we have the logarithm of the maximum plasma concentration, or Cmax. So as you're moving from left to right, you have higher exposure in patients. And as you can see on the distribution in the margin there, we have a good separation between toxic compounds having higher exposure and non-toxic compounds having a lower exposure in patients. Now, if you look at the y-axis, that is the point of departure, the logarithm of the point of departure in the assay. So if you move up, you get more potent in the assay, and we've normalized it to the top tested concentration. So the top tested concentration, if you're inactive, and that's 316 micromolar, then you get a value of 0, and then 4 logs from that top tested concentration is the maximum potency that you would achieve in the assay. To train our model, we have split the data into 65% training data set and a 35% test data set, and we've upsampled the train data set to balance the two classes together. Now, in most other talks, you would see now calculated these different safety margins, but here I'm trying to argue against this approach. On the face of it, using a safety margin seems like a good idea. I've used the approach before, it's well cited in the literature, and it makes intuitive sense. You're linking a hazard estimate in the form of an IC50 or a lowest effective concentration with exposure in patients, the maximum plasma concentration, to get an estimate of the risk for those patients. However, what happens next is that you have a list of compounds where you determine the optimal threshold to separate the inactives from the actives based on a ROC curve. That approach is prone to overfitting, doesn't generalize well to new compounds, and it doesn't give you any way of incorporating the uncertainty in your IC50 determination and your Cmax determination. Moreover, if you use this approach in multiple assays, you introduce correlations in previously uncorrelated assays, which could lead to problems when you're trying to fit models later on. You're also overestimating the importance of Cmax by incorporating it in multiple predictors. You finally get a less flexible binary classification. You're either non-toxic if you're just below the threshold, or you're toxic if you're just above the threshold, but you get no probability, no sense of a proper way of ranking compounds. That kind of classification is detrimental to good decision making. We don't need to be giving projects labels for their compounds, whether they're good or bad. We need to give them a probability of there being a safety liability for this compound. This will enable them to make the right judgment based on the utility function that they have. For example, if you're based in oncology and you're working on a life-saving drug with no alternative, you would be able to tolerate a high level of risk for that compound compared to a compound for diabetes or for a long-term treatment in a cardiovascular disease. Similarly, we need to move away from these classification type metrics like sensitivity, specificity, and accuracy towards more probabilistic scoring metrics. Frank Harrell has a really nice blog on why we should move away from classification metrics to probabilistic metrics, but just as a taster, I mean, they turn the probability the wrong way around. For example, sensitivity gives you the probability that you're going to score active in the assay if you already know you're active. But what projects are normally interested in is, when we score active in the assay, what's the probability of there being a safety liability? And that is the correct way to look at it. The Brier score is a much more appropriate metric to score how good our predictions are. The problem with the classical Brier score is that it's difficult to understand it in perspective. To put it in perspective, we need to compare it to something. And this is where the Brier skill score comes up, of where you're comparing the Brier score from your prediction to one that is informed by either a historical average or the class proportion. So the Brier skill score would range from -infinity to completely worthless, to 0, where you have your unskilled forecast based on historical averages, and 1, you being a perfect forecaster. Nate Silva, you may know him from his blog 538. He also has a really good book on predictions called the Signal and the Noise. And on their blog, they give the Brier skill score for their predictions in different domains. For example, their predictions in sports forecasts or in politics. And as you can see on the graph here, they're much closer to the unskilled forecasts for sports events than they are for politics. So for politics, they're giving better predictions. And I've highlighted here their predictions for the World Cup matches in men, which is basically where we are with our predictions from in vitro assays extrapolating to the risk for causing drug-induced liver injury in patients, for example. Okay, so let's go back to our data set and some basic truths in toxicology. We know that as we increase patient exposure to compounds, we have a higher risk of getting side effects. And the same is true for the drug-induced liver injury data set. So there are many ways that we can find the threshold that separates these two sets of compounds, the actives and the inactives, and that we get a classification. But what we would like to do is we would like to do something better. We would like to get a probabilistic classifier. And here I would like to show you a very simple way to begin, and that's with a logistic regression. It's very easy to fit it, so that's why I thought I'd illustrate it with an example in R. You basically call the GLM function, and you say that your probability of getting drug-induced liver injury is proportional to the Cmax. You use a binomial distribution because there's only two classes, toxic or non-toxic, and we use the upsample training data to get our model. What we then got is this logistic transformation where we have the log odds of the probability of causing DILI versus divided by 1 minus the probability causing DILI, and a linear model which includes the Cmax. And that's already big help because we don't just get a binary toxic versus non-toxic distinction, but we get a probability estimates. Then that allows us to say, okay, how much better are we with this model just using Cmax than just guessing the probability of DILI corresponding to the class distribution. So we have 63% of our compounds causing DILI in our training data set. So if we were always to call probability of 0.63, what will we get? And that will be a 0. What we can see here is that we're better than this. Our Breer skill score is 0.16, and there's 243 compounds that we predict better than if we would have used just the distribution of the classes. Now, what happens when you introduce the potency in the imaging assay represented on the y-axis? For the model, it's really simple. You just basically get another extra term, which is your PIC50 in phenospace assay, and you get another term in the equation as well. In terms of your skills, the skills are improving, so we're moving further away from 0 and closer to 1. We now have a median Breer skill score of 0.21, and we have 11 extra compounds that we're predicting better than we would have otherwise predicted by chance. Now let's compare the two classifiers. With the vertical line, we have the classifier relying solely on logarithm of Cmax, whereas the sloping line represents the classifier that uses both Cmax and potency in the assay information. And the extra value that you get from the second classifier that uses potency in the assay is denoted by these two triangles. There's an upper triangle where we have compounds with intermediate and high potency in the imaging assay, but fairly low Cmax. And in the lower triangle, we have compounds which have a low potency in the assay or are completely inactive in the assay, but have a slightly higher Cmax that would have otherwise classified them as DILI positive. And this is where the difference between the two classifiers comes in. If you're using the classical boundaries of 50% probability of DILI classifying a compound into the toxic or non-toxic class, what you get is a net gain of 19 DILI compounds and a net loss of about 5 no DILI compounds when you're using the information from the imaging assay. The benefit here is that we have used a probabilistic predictive model that doesn't just separate the compounds into active and inactive, but also gives us the probability of them causing drug-induced liver injury. So we can see in a plot like this, when we've plotted all of our compounds on the x-axis and their probability of causing drug-induced liver injury, is whether the blue compounds cluster below a certain threshold and whether the red compounds cluster above a certain threshold. I've illustrated here with the classical threshold of 50% probability, but you don't need to stick to that threshold. Depending on the project, for example, if it's an oncology project, you may be able to tolerate a high level of risk, or if it's a cardiovascular project, you should be more conservative. You can change that boundary. The other bonus that you get is that you can compare the individual predictions and how good they are between different models and see where the differences are coming from. And this is what I've done on the plot here. You can see that in addition to the point colors that we have, each individual point receives a halo. And the halo is meant to represent the scale difference between the classifier using imaging information and the classifier that just relies on Cmax. Now, if the color is gold, and that means a scaled skill score of about 1, we're making much better predictions. And if the color approaches purple, then we're making worse predictions, as in the case of paclitaxel. And what you can see is that we have genotoxic and cytotoxic compounds such as paclitaxel, docetaxel, the vinca alkaloids, and colchicine being receiving worse predictions compared to if you were just using Cmax. What that means is that we have assay interference, and our imaging assay is picking up in-specific cytotoxic and genotoxic compounds in addition to the specific hepatotoxic signals. What we've known for a while is that you can't rely on a single in vitro assay in cells to predict the complex response that you're going to get in humans, the different mechanisms of action of the different drugs and the different ways that the drugs can be toxic. That's why we have used different approaches which combine multiple cell lines and multiple assays. And in all of these approaches that we can sum up as the bin and sum approach, you always have some form of exposure on the x-axis, and then on the y-axis you have different varieties of these arbitrary scores derived from binarizing or dichotomizing the results that you get in the different assays. And Stan Lazick and Dominic Williams do a really good job at reviewing all the problems that are associated with this approach, but I'll try and summarize them here. First of all, what you have is you have multiple assays where you arbitrarily call something active or inactive according to a threshold. All the assays in this approach are usually given equal weight and you sum them up. You don't capture any interactions, and if a compound registers weakly at the threshold or just below the thresholds in multiple assay, that is not properly captured as well. So the bin and sum approach is not a very sophisticated and not the preferred approach of combining multiple different assays. In AstraZeneca, we have now moved on from the bin and sum approach to exclusively using Bayesian probabilistic models when we want to predict from in vitro assays risk for humans. And I'm going to illustrate it with a couple of examples that have been recently published in the literature, which show the value we bring when you use those probabilistic models. They both use a finer distinction for drug-induced liver injury. If you remember before, we had toxic and non-toxic, but now we're introducing a third class. So we have low, medium, and high probability at causing drug-induced liver injury. We've also used two different models. One is an ordered logistic regression, and then the other one employs a Bayesian neural network to try and predict drug-induced liver injury in humans. We've used compound properties such as lipophilicity, the Cmax that they achieve, as well as assay results like BSEP, the Glugall assay, and standard cytotoxicity assays as predictors. What we're getting with these Bayesian predictive models is better predictions and finer distinctions between the classes. For example, on the left we have the ordinal regression, and as you can see, we're already predicting the DILI severity according to three classes, and the compounds are separating by class. With the Bayesian neural network in the middle, this distinction is even more pronounced, and the classes are separated better. Finally, the plot on the right is showing the Breyer skill score. If you remember from the previous slide, 0 was the score we're going to get if we are always making the historical average or the proportional, if you're guessing probability equal to the proportional distribution of the classes. What we see here is the most of the area is above 0. And if you compare it to the results we were getting with the imaging assay, we were getting a Breyer skill score of 0.21. Here we're getting Breyer skill scores in the realm of 0.35, 0.37. So as you can see, a gradual improvement, better predictions, and finer distinction between the classes. Incorporating results from different assays brings the promise of better, more predictive models, but also carries with it certain challenges. For example, we don't always understand the biological link between an assay result X and the clinical outcome we're trying to predict in Y. In the absence of strong biological and theoretical knowledge, we're left with a choice of which function do we use. And as you can see on the panels on the right, the choice can be quite overwhelming. We could choose a quadratic, 2-parameter exponential, or a 3-parameter exponential. What we could then look is, okay, what is the sum of all models? How do all models look next to each other? And look at the uncertainty that is associated with the model averaged function. What that would help us in, as you can see in panel F, is we can understand what is the domain of the assay results, in which values for assay result in X do we have agreement between all the possible models that we can use, and where do we start to see disagreement? Where is the point where our certainty is decreased? Where is the applicability domain that we could use this assay in? As the biological complexity of the assays that we're trying to study increases, we have to employ more sophisticated models to describe that biological complexity. And a good example for that is our studies in protein degradation, where we have these bifunctional molecules called PROTACs, or protein degraders. What they have is one part of the molecule which reacts with the target of interest and doesn't necessarily inhibit it, and another part of a molecule which tags the protein for degradation. Now, what is characteristics in those responses of protein degraders is that they're not monotonic. There is a point where the complex is saturated and where we get this so-called hook effect. So when researchers are trying to fit a normal 4-parameter curve fit model, we get unreliable potency estimate, we get a lot of masking that needs to be manually done, and the rankings for compounds are unreliable. That's why we have chosen to quantify that uncertainty and model those responses with Gaussian processes so that we can fit better responses and we can get more reliable ranking in compounds incorporating uncertainty. This is an example of how the whole process works in practice. We have two main panels here. A panel on the left, which shows us the potency of the compounds along with the uncertainty estimates, and a panel on the right that shows us the ranking when you draw multiple draws from those distributions. Now, for some compounds, which are either really potent or really weak, the ranking is very stable. They're well separated from everything else, and as you can see, they always rank at the same rank. Other compounds, in contrast, are less well separated, and there will be some variability in the ranking. That gives projects a much better view as to how stable the rankings are and how well we can separate different responses. That enables them to make better decisions and prioritize compounds for development. In conclusion, I hope I have managed to convince you that in order to make good decisions for the benefit of patients, we need to be using probabilistic models which account for uncertainty. Those are the kind of models that enable human judgment and make the most use of the evidence provided. And those are the models that we should be using in our toxicological studies. As a scientist who develops and implements in vitro assays, this has been a real journey for me, and I wouldn't have been able to do it without the help of my colleagues and collaborators. I would like to call out Jasmine Sandler, who has really contributed into the development of the imaging assay, Adam Corrigan, who's developed a neural network classifier for that, Elisaveta and Avid for pioneering Bayesian approaches and demystifying them for us, Dominic for his leadership and views on the implementation of Bayesian approaches in the DILI field, Stan Lazik, who has been leading the thinking behind this and the strategy and is now joined Prioris AI, and of course our colleagues and collaborators from Unilever who have helped us in our exploration of Gaussian processes. I encourage you to screenshot these references because these are some of the papers that have influenced my thinking and have convinced me that using probabilistic predictive models is the right thing to do in our toxicological studies. Please find me on LinkedIn and let's talk about drug discovery, the right models to use, the right assays, and the tools that enable good decision making.